Okay, so lecture 4 we start to go into some specific topic. So we start with bacteria. So we start with microbial biotechnology, and later we will move on to plants, to animal, and at the end like a summary on everything. Okay, so bacteria first. So using bacteria, using microbial biotechnology, we demonstrate the steps of doing recombinant DNA technology. So how to make the recombinant organism. So again, we start with DNA isolation, and that's why you have to know about DNA isolation because that's the very first step. You isolate the DNA, but first you have to be able to identify the DNA you want for a certain characteristic, like producing insulin. You have to know which genes is producing the insulin first. But then you can do DNA isolation. You isolate the part of gene you want, the gene of interest or the target DNA. And then you can do molecular cloning, or you can do PCR to make copy of them. And if you do so, you have to run a gel. I'm still following this. You have to run a gel to check whether the gene is really amplified. You have enough of the materials and that you can work with. Okay, so this part is on isolating the DNA, blah blah blah, and then amplifying the DNA, and that's why PCR happen again. Okay, so for PCR, you have to, I think you're very familiar with them already. You have to know about like the raw materials, the thing you mix together, what are they? Each of their function, why you have to add them into your master mix of PCR. And we have 3 steps, okay? So don't mix it up with the transcription and translation 3 step. But still you have to be able to tell the process of PCR, because although they're so commonly used that you don't think they're special, but they're so important. If you don't have the PCR, you don't have the DNA amplification, then you will not have enough DNA materials to work with. No matter you want to make a recombinant organism or you want to just check the DNA, you first have to be able to amplify them so that you have enough of the DNA to work with. Okay, so make sure you know the steps, the process, the raw materials, the principle of doing the PCR. Okay, after PCR, you have to check whether it is successful or not. So we come back to gel electrophorescence. So also basic principle on how a gel electrophorescence work, why they can like separate your products based on what property, so that and you can visualize them to tell the molecular weight, the molecular size, the length of the DNA. Okay. And then we talk about the cloning factor. The cloning factor that allowed you to make the copy into still the DNA. And we use E. coli a lot. And we have some other choice, basically E. coli. Okay, so some example here, just some possible available factor you can choose with. And we use plasmid, which is a very useful cloning factors. And the alpha complementation actually is the same idea of the blue-white screening. Okay, so this is for you to check whether the plasmid are incorporated successfully or not. So again, color change, colony a lot. If they are small colonies, what are they? So these are the basic idea of checking the result of a blue-white screening. And then transformation again. Okay, for bacteria, we have many methods. Again, you can do heat shock, you can do electroporation, and you have to allow them to recover a bit after transformation, because the cell are kind of fragile after transformation. They are forced to open up more. They are protecting layer, so you have to allow them to recover before you further grow them. Otherwise, they may just die after this stage. You have to give them some break here. Okay, he saw electroporation, recovery, and then hopefully the DNA will get inside and also incorporate into the genome or as a separate plasmid, but they can still do it. Okay, and spec plate, you have really do it by yourself in the lab. Okay, so those are the basic steps. If you are doing recombinant bacteria, you have tried it yourself, so you should be very familiar with each step point to take care of, and what will go on if you did something not exactly the same following the procedure. And then the second part will be some application of microbial biotechnology. So in many cases, we use the recombinant bacteria for biosynthesis because they grow fast, so they produce the protein products very fast, and we can collect them. And they are like single cells, so they're easy to like extract, to grow, and to purify to get your products. Okay, so some example of biosynthesis, we can use them to make ethanol. Okay, so you don't need to go really detailed on everything, but at least you have to be able to tell some example of such application. Like they can be used to make ethanol production. We can modify something so that it can grow. It can have a faster metabolism. They can produce faster, or they can make use of raw material originally they cannot use because we change their gene. So some brief explanation like that to illustrate how the microbial biotechnology can help speeding up production of something. Okay, so we have example of ethanol. We can modify some yeast or bacteria so that they can use more type of raw materials and therefore have a higher application in the industrial ethanol production. We also can use them to make antibiotics, so still ethanol here, ethanol. And then we can use them to also make antibiotics, and we can also use them to make food additives and other type of food production, mainly the fermented foods, because we already use this mycobacteria to make fermented food like yogurt, cheese. Okay, so make sure so many daily life, real life application here. Make sure you can name a few if you're asked to.